Hello and a very warm welcome. Dr. Abhishek here, and in this video we are continuing with our theme of how to score well, rather how to score very high in your main examination. What we are doing in this video is that we have taken a question that has come in the main examination recently, and we are going to analyze that question and we are going to together build a good model answer that enables you to get high score from the examiner. Before starting, let me remind you as always that high score is never given to you. High score is never given to the subject. High score is always fetched by the quality of your answer. So if the examiner is happy with the quality of your answer, examiner is bound to reward you very well. At least in subjects like botany and zoology, there is a clear cause and effect relation between the quality of answer and the score that you are eventually getting. So the question that I have taken for today's discussion is from chromosome packaging. This is a topic that finds a mention both in botany and zoology syllabus. It is a part of paper 2 under genetics section. In zoology, the question on chromosome packing is framed like describe the molecular structure of chromosome or describe the difference between euchromatin and heterochromatin. How is the chromosomal material packaged within the cell? So those kind of questions have already come in the examination. And in botany, the question was asked this year only in 2021 main examination, describe the types and composition of chromatin. How is DNA packaged in a eukaryotic cell? To make it very clear to the candidates, the examiner has already divided this question into two parts, 10 plus 10. So it is very obvious that you have to give equal weightage to both the queries. Now, before proceeding with the answer writing, let us identify the key query words, the key words of the question. So the keywords are already put in the box. So first keyword is types of chromatin. Second keyword is composition of chromatin. Third keyword, chromatin itself, because this is where ideally your answer should start, where you define chromatin and explain a bit about chromatin. The second part of the question demands you to know about the DNA packaging within the eukaryotic cell. So one thing is very clear that the first three queries, types, composition and chromatin, we have to write for 10 marks. And the second part of the query, where we have to simply talk about how is DNA packaged in a eukaryotic cell, that we have to write for 10 marks. In the UPSC main examination, for every 10 marks, we are supposed to write about 120 words. So 120 words essentially mean 12 sentences to 14 sentences, depending on how long a sentence you are habituated of writing. Most of us write sentences between 7 to 8 words. Sometimes a longer sentence can go up to 10 to 11 words. I will highly recommend that you should not be writing very long sentences. Whenever you have more than one ideas which you want to bring into one sentence, it is always better to put them into two separate sentences. They are always easier for the examiner to read, and the easier it is for the examiner to read your answer, the easier it will be for the examiner to assess the quality of your answer and reward you appropriately. Complex writing has absolutely no place in the UPSC main examination, be it your optional subject or general studies. So now you have understood the query areas, and now you also understand that this entire answer has to be written in approximately 240 words, and out of the 240 words, close to 120 words will go for the first part, types, composition and basic about chromatin, and the second part will be containing 120 words, but this will be on how DNA is packaged in a eukaryotic cell. Okay. Now, sometimes you will need to reorder the sequence of the keywords. For example, here it is given types and composition of chromatin. But if you notice, your answer should ideally start with what is chromatin and giving the examiner a basic idea, basic introduction of chromatin. After that, before coming to types, you should be coming to the composition of chromatin, because in typification of chromatin, composition has to be mentioned. So it is always logical to bring composition before type of chromatin. And once you are done with this, then you can come to the packaging of DNA in the eukaryotic cell. So one lesson that you should get from here is that sometimes you will need to reorder the keywords. You do not need to follow the sequence of keywords as it is given in the question. Sometimes it is so logically laid out already that you can very well follow the sequence. Sometimes it may not be the case. When it is not the case, you will have to reorder. So here we have reordered. So in this answer, I am going to first write about chromatin. Then I'm going to write about its composition, then types. Then I will come to the second part, how is DNA packaged in a eukaryotic cell. Chromatin, I will give approximately 3 points. Composition, approximately 6. Types, approximately 6. Here, approximately 10 points. So that is how, so approximately 25 unique points I'm going to use in this answer. And later during the construction of the answer, you will be noticing that how we are trying to combine a specificity, some unique facts without going into excessive detail, because excessive detailing is certainly not expected by the UPSC. And also you are not going to retain all of this to your examination hall. But whatever limited you can understand, whatever limited you can retain, how to use those things to your best advantage is what we are going to discover in the later part. Okay. So now let us see, we are ready with chromatin. 3 points I am going to mention about chromatin. First and foremost, my point is that in the eukaryotes, the nuclear DNA is organized into chromatin. Now, if you see, I have underlined nuclear DNA is organized. Why? Because in the eukaryotic cell, apart from the nucleus, even mitochondria and chloroplast can contain DNA. But the point is that in the mitochondria and chloroplast, the DNA that is present, that never gets organized as chromatin. It is only the nuclear DNA that will be getting organized as chromatin. A small fact, obvious fact, but it needs to be told to the examiner. Remember, the examiner knows everything. Only thing is the examiner wants to hear it from you. So if this is a specific point that you can underline, that you can kind of highlight, you must do it, because such attention to detail is bound to create a very positive impression of yours in the examiner's mind. And this positive impression can actually secure you a little better marks than most of your competitors who do not pay attention to detail. And please understand, when we talk about attention to detail, we do not mean that you should go very deep. Going very deep is certainly not a feasible idea when we are dealing with a lot of topics within the syllabus. So the depth has to be always optimal, as I have discussed in my previous video. But within that depth, whatever you have retained, learn to play with the facts to create maximum impact in your favor. Play with the facts to create maximum impact in your favor. That is what I am doing here. All of us know that mitochondria have DNA, chloroplast have DNA, but this DNA is not organized into chromatin. Nuclear DNA is organized into chromatin. After that, if you want to write a little more, you can write that chromatin is uncoiled during the interphase and chromatin is coiled during the M or the dividing phase. Next point about chromatin is going to be that chromatin is a nucleoprotein complex, and again paying attention to some specific information, this nucleoprotein complex is going to have a single DNA which is double-stranded and histone and a few non-histone protein. But emphasizing the fact that each chromatin is going to contain a single DNA is again kind of underlining what may seem obvious to you, but a lot of your competitor are not going to kind of highlight this small specific fact, though they are knowing it as well as you are knowing, but knowing is not enough, showing is needed. Oh, so that is a nice one-liner. Knowing is not enough, showing is important. So yes, if you know it, you show it. Okay. So you have to keep in mind that yes, this is something most of my competitor may not be writing. So negligence can cost them and attentiveness will pay you. Okay. Then chromatin is organized as 30 nanometer fiber. That is something you should mention in the introduction. In the older days, we used to call chromatin a solenoid structure. Then the time passed and scientists gathered the evidence that it is not solenoid. So we started calling it semi-solenoid. Then we started calling it the zigzag structure. But today, the molecular biologists and author, they simply call it 30 nanometer fiber. So it is a 30 nanometer fiber level packing of nucleosome. So chromatin is a nucleoprotein, single double-stranded linear DNA per chromatin plus histones and some non-histones. It is made up of only nuclear DNA and it is in the form of 30 nanometer fiber. So these are the basic things. Now we are ready to do its composition. So first thing, again very obvious, that a chromatin will carry equal amount of DNA and proteins. DNA पे एक ही सेंटेंस लिखना काफी है कि that this DNA is linear, double-stranded and one per chromatin. That's it. And then there are two classes of proteins. Histones are the major proteins. Histones are present in all eukaryotes except in a group of algae that they are known as dinoflagellates. And for this reason, dinoflagellates have been called mesokaryotes. If you have read an Indian textbook on these topics, you will get this information. So sometimes it is important to combine foreign author with an Indian textbook. Foreign author will give you the cutting edge latest information. On the other hand, Indian authors will give you something unique that foreign authors may end up skipping. So here is the fact. Then some of the core information about histones, that they are low molecular weight protein. Be specific. Low molecular weight, someone else is writing and you are writing low molecular weight, 11 to 23 kilodalton. Who do you think is going to get more marks? What do you think? Is it very difficult to remember? Answer is no. If you have paid attention, you will be able to remember. A lot of students who score well, who get selected, they remember these tiny little details. So if you remember, it is going to pay you very rich dividends. They are rich in lysine or arginine. लोग carelessly क्या करते हैं? They simply write these proteins are rich in lysine and arginine. No. Lysine and arginine नहीं होगा. It is either lysine rich or it is arginine rich. These small little details. Okay. They are basic. That means they have positively charged surface, and that is how it binds to DNA electrostatically. So a lot of people will write it will bind to DNA. You are going to write it is going to bind electrostatically, right? So I used to have a senior during my master's who secured a very high rank in the civil service examination. So since she is a serving officer, so it would not be appropriate taking her name. So I'm not taking her name. But she taught me this art of using even the obvious information to the best of one's advantage. And that is what I'm sharing with you. So when everyone is writing that histone attaches to DNA, you are writing histone attaches to DNA electrostatically. Why? Because histone has lysine or arginine richness, because of which histone surface is positively charged, and the DNA, because of the phosphoryl group residue, DNA is going to have surface negative charge. Okay. Then 5 types of histones. What we have done, we have put the molecular weight. You may like to dismiss right in the beginning that, okay, who remembers the molecular weight? My point is, this is the only category of protein for which you are supposed to remember the molecular weight. There is no other protein apart from histones and nuclear pore complexes for which you will be needing to remember the molecular mass. So if there are only 5 proteins plus 1 nuclear pore complex elsewhere in the syllabus, if only these 6 molecular masses are supposed to be remembered, why not? We can remember it. Histone 1 is 23 kilodalton. Histone 2A and 2B are very close to 14 kilodalton. Then histone 3 is 15.3 kilodalton and histone 4 is the smallest, 11.3 kilodalton. Okay. The first one is lysine rich. The second and third one, that is H2A and H2B, are slightly lysine rich. Okay. And the last two, H3 and H4, are arginine rich. So top one, lysine rich, 2A, 2B, slightly lysine rich, 3 and 4 are arginine rich. ये बहुत मुश्किल नहीं है, but if you put it, it will instantly separate you from 98% of the candidates. Some of you, I'm knowing that you must be nodding your head in disagreement कि नहीं नहीं इतना याद नहीं होगा. That is your take. आप मत लिखो. It is a free country and you have total freedom of expression. What do you want to write? But as a teacher, I will want to still insist that this is what you should do. Whether you do it or not, it is entirely up to you. But yes, if you do it, you will yourself feel the benefit, and this is going to be an attitude. Even this year, I was meeting a candidate who's already in the revenue service and wanting to improve his rank with the same subject, zoology. So I was noticing his self-made notes, and I was really impressed what an attention to detail this candidate had paid. So those who get selected, those who score high, have an eye for certain detail. And these are not, again, detail does not mean depth. It only means a reasonable level of knowledge, but then this eagerness to remember and retain this knowledge and use it to the best of your advantage. Then non-histone protein, 2 cohesins, which keep the sister chromatids together after the S phase, and condensins, which will be coming into play in the M, that is mitotic phase, and they will be condensing the chromosome into the M phase chromosome. So well, that is composition. We are done. We don't have any more point. Now we are ready to write the types of chromatin. So whenever you are writing the type, you have to tell the examiner on what basis am I identifying the types. So I'm writing 2 major types depending on relative condensation during the interphase. So you have got euchromatin, that is uncoiled. So ये वाला part euchromatin है, देख रहे हो, ये uncoiled है during the interphase. It is simple 30 nanometer fiber. There is no other protein other than histone. It has got more genes. Apart from that, it is showing greater transcriptional activity, and it also replicates earlier. On the other hand, if you notice, this is heterochromatin, this is heterochromatin. So what happens that it is coiled even during interphase, while euchromatin is not coiled during the interphase. Euchromatin will coil, but during the M phase, not during the interphase. But heterochromatin remains coiled even during the interphase. It is more condensed, and because of this, it appears dark since it is a condensed form. It has less genes, low transcription, and it has late replication. These are the types. We are done with the first part of the answer. Moving next, how is DNA packaged in a eukaryotic cell? Now here what I have done, that I have created a subheading which I am kind of knowing that my competitors are not going to write. That is the need. Why do we need to package DNA at all? पहली बात तो यही होती है, and it needs to be packaged because the total length of DNA is much more than the volume of the nucleus. DNA की जो total length होती है is much greater, thousands of times greater than the volume of the nucleus. So अगर हम nucleus की बात करें ना, तो it is just 5 to 10 micrometer in human beings. Okay. On the other hand, in the human beings, the total 46 chromosome, the DNA that they contain, if we extend this DNA, it will be 2 meters. So 5 micrometer. What is micrometer? 10 to the power minus 6 is micrometer. Right? And this is 2 meter. So do you get it? It is the difference of how much? 2 million. Right? It is a difference of so much. So therefore, the DNA needs to be packaged. Okay. Now, how is DNA packaged? So DNA is, so after need, we are coming to the mode. So DNA packed with structural proteins, and how? Hierarchically. So these are some of the things. Means step by step packing होगी. पहले DNA एक सेट ऑफ प्रोटीन के साथ बाइंड करेगा, फिर एक स्ट्रक्चर बनाएगा, फिर उसके बाद के लेवल की कंडेंसेशन होगी, फिर उसके बाद के लेवल की कंडेंसेशन होगी. So this binding is hierarchical to form the completed compacted structure called chromatin. The extent of this condensation is known as packing ratio. What is packing ratio? So I have given yet another important information, that is total length of DNA upon the length of the M phase chromosome. So for example, for a human chromosome number 2, so 14,000 micrometer is the extended length and 2 micrometer is the compacted length. So what is the packing ratio? Packing ratio is 7,000. So once we have mentioned this, then the only thing that we have to do is the hierarchy. So hierarchy, I have divided into 3 columns. That what is the structure, what is the thickness at that level, and in which stage of the cell cycle does it occur. So first level is naked DNA. Thickness is just 2 nanometer. It occurs during the synthesis phase, क्योंकि synthesis के समय DNA is made totally clear of the structural protein, then only it can be replicated. So naked DNA you will find only in the S phase. The moment DNA is replicated, histone proteins will bind and they will form nucleosome, the thickness of which will be 11 nanometer. And this is something you see in late part of S phase. So it is not S phase, it is the late part of S phase. After that, some non-histone will bind and histone tail will link with each other, and that will make uncoiled 30 nanometer chromatin fiber. So this 30 nanometer chromatin fiber will be uncoiled one, normal chromatin fiber. Okay, 30 nanometer. So for most of the interphase, it is the 30 nanometer chromatin fiber that will be existing for most of the interphase. And then as the cell progresses to the M phase, then more non-histone proteins, especially what? Condensins. So these names have to be mentioned. So especially condensins will attach, and then coiled chromatin will be formed that we know as M phase chromosome. What are the dimensions? The thickness is 700 nanometer for one chromatid, and this is found in the M, that is the dividing phase of the cell. After that, you make the diagram. So 2 nanometer naked DNA, there is step 1 where histone proteins attach, and this is the nucleosomal structure that forms. After that, the nucleosomal structures, they coil around their own axis and they make 30 nanometer chromatin, and then that will further coil and that will make a M phase chromosome where one chromatid is 700 nanometer and both the chromatids together will be 1400 nanometer. I have purposefully made this diagram with my hand so that you have a realistic idea that okay, this is what we can draw. Okay, तो बस इतना ही आप draw कर सकते हो. So I hope you understand. Yes, your diagram can certainly be better than me, but this is the minimum standard I will be expecting. If you make better without violating the time limit, I will be the happiest person, right? So this much. Now if you have time, ऐसे तो आपका आंसर यहाँ पे फिनिश हो जाना चाहिए, and after this you should be writing the conclusion, but if you have time, you can explain step 1, step 2 and step 3. So naked DNA just after replication, electrostatically bound by H2A, H2B, S3, H4, 2 copies of each, then that makes nucleosome core, 147 base pair of DNA, 8 histones, then bound by histone 1, then a chromatosome is formed, 166 plus minus 2 base pairs of DNA, and now histone 1 आ गया है तो 9 histones. That is the nucleosomal level. Then binding between histone tails and linker DNA plus chromatosome linking. You don't have to show the diagram because you are after all writing a short note. So that is 30 nanometer chromatin fiber. Formerly we used to call it solenoid fiber. क्यों लिखा formerly? Because if the examiner is expecting कि अरे बच्चे ने अभी तक solenoid नहीं mention किया, mention कर दो, right? Then binding of condensins. That will form looped chromosome structure. No need to draw the figure here. And that makes condensed M phase structure. Now it is really the end of the answer, and you can write the conclusion that chromosome packaging is correlated to genetic and cellular function. How? It allows gene expression and repair during the interphase because उस समय ये loosely packaged होता है, and it allows damage free division during the M phase जब ये condense होता है. So when it is uncoiled, it allows gene expression and it allows repair And when it is coiled, then it prevents chromosomal damage and it allows the sister chromatids to be separated. And upon this separation, it will be going to the daughter cells. So, well, that is the end of our answer. And once we are at the end of the answer, let us remind ourselves the basic thing: high score is never given by the examiner. Examiner is not duty bound to give you high score. Get out of this notion that your subject is scoring or non-scoring. Even if subject is scoring, without a good answer, you're not going to get good score, plain and simple. So you are not entitled to get high marks from the examiner. Examiner is not obliged to give you high marks. The only way you get high marks, question after question, is through great answers. Great answers do not need to be very deep in their knowledge base. Only thing is that the knowledge that you possess, you need to know what are the things which you can do with that knowledge only, which your competitors are unlikely to do. Mind it, it is not only a competitive examination, but it is one of the most fiercely competitive examination, not only in our country, but perhaps in rest of the world also. There is hardly any competitive examination that matches the civil service examination in its depth, in its rigor, in its number of papers, and the sheer competition. So you have to be at the top of your game, and being at the top of the game will have answer writing as a very important component. But before you embark upon answer writing, you need to be in very solid command of the facts, because you will have to play with these facts to write emphatic answers. So I hope you found this video as well adding value to your preparation. I will be coming up with many such videos in future also. So in the meantime, please practice along these lines, because great answers are never built in the pressure of the examination hall. They are built when you are still preparing, and then you will retain these answer frameworks, the key facts, the key concepts in your mind, and then you will be reproducing it in the examination hall within the time limits. So that is all in this video. You take a very good care of yourself, your preparation, and I'll see you soon. Goodbye and good luck to all of you. Goodbye.